are looking at different angles how to deal with different diseases. That's why we can see even new ways of looking at old disease. As one of the speakers will be speaking, and I do hope that this webinar will, as I say, always ignite the young minds to take this particular webinar seriously, and also maybe take research as their field in the future, not just to complete their BSc, but then not only to complete their masters, but take up something, a path that will take them to very high, deeper kind of research. So today, as we assemble here and as we participate in this webinar, I wish all the best to the organizers, and wish that all those participants, all those viewers will really benefit from this particular webinar. And thank to all the speakers who have consented to come and share the knowledge that they have. So thank you once again, and welcome to this particular webinar, and all the very best. Thank you.
Thank you, Father. Thank you, Father. And now I would like to ask Assistant Professor Dr. Tamal Majumdar to please introduce our speaker.
Hello, thank you, Aranda. Good afternoon, Father Dr. Lalit Tirki, SJ, Principal, North Bengal St. Xavier's College, Dr. Devasri Datta, Scientist, Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram, my dear colleagues, my dear students, and all the participants here in this particular webinar. It's a great pleasure for me, in fact, to introduce Dr. Datta to the assembly over here. She is a persona with a lot of potentials. You can unravel with her speech, certainly, that we'll do certainly in a short time. But just to go through what she has achieved so far is really fascinating to just to discuss, she initially had done her BSc in chemistry from University of Calcutta, then masters in biochemistry from University of Kalyani. She did her MTech in life sciences and biotechnology from Jadavpur University. Then she obtained her PhD from Jadavpur University, and then for a postdoctoral research, she went to University of Kansas Medical Center, USA. Her publication list is very interesting and something to be proud of. She has already got 19 papers in all internationally acclaimed journals that include like those of PNAS, Proceedings of Natural Academy of Sciences, Journal of Biological Chemistry, Journal of Cell Science, Stem Cells, Molecular Cancer, Scientific Reports, etc. So that is enormous. She has already got one patent in her name from the stem cell biology, a US patent it is. She has received several major awards and honors. DBT National Women Bioscientist Award 2016, Award of India International Science Festival, Women Enclave Lucknow 2018, EMBO Travel Award for delivering talk at EMBO workshop held in Greece in 2019, a DST Fast Track Fellowship in 2013, American Heart Postdoctoral Award in 2011. So from here only we can really just think of, we can imagine what her potentials are really. She has got certain other achievements as well. She is serving as RGCB, this Rajiv Gandhi Center for Biotechnology Master's Program in Biotechnology, as a course coordinator since the very beginning. She has already had 2 students PhD from her, finished PhD from her. One is doing one of them is doing postdoctoral research in Riken, Japan, and another one at the University of Wisconsin in USA. She has already received 8 grants, means projects, from national funding agencies like DBT, DST, CSIR, and SERB. So, to say the least, I'm sure that we will be enlightened a lot by Dr. Datta. So I welcome Dr. Datta for the speech. Ma'am, it's yours now.
Thank you, Dr. Tamal, and it was really a very kind introduction for me. And good afternoon, Principal Sir, all the teachers, and very prominently the students of the college. And in the beginning itself, I would like to say that I have prepared the talk in taking the perspective that the students are the majority of them, and which I think I could see from their list, like third year, fifth semester, fourth semester, and all this. So I have taken their point into consideration, and I have prepared my presentation like that. So good afternoon to all of you. And are my slides visible? Is it visible? The slides are visible?
Yeah, now it is. Now it is. Yeah.
Okay. Thank you. Just a second. Yeah. So today's talk is based on induced pluripotent stem cells, a classic example of union of basic and translational research. Now, why I'm talking about basic and translational, you know, there is always a competition, or there is a confrontation, I would say, between different researchers. I mean, you all will be also become researchers. I hope so, at least many of you, that I am doing the better science because I'm working on basic science, and somebody will tell what you are going to do, because until unless there is translation, there is medicine, there is drugs to the disease, nothing is impossible. So that is translational science. But both of them depend on one another. If we have not studied what is the structure of DNA, which is actually basic science, there would not have been possible to design so many drugs, because there is minor groove, there is major groove. You know that this particular thing binds to a particular this groove or not. Then all the antibiotics, how do they act? This is all part of research. So finally, you are putting the basic science, which scientists are doing for the huge period of time, and then you're putting all this knowledge to be transferred into the translation research, which is ultimate for the benefit of all the human beings in the earth. Okay, so before I start this one, I would just like to ask that, as far as I know, the students who have joined here are from zoology, botany, and microbiology. I was informed like that. So my question will be, like, is it like, since it's a webinar being organized by your department or your college, you are appearing for the webinar? Is it like that? So why should you actually know about stem cells? Is botany people, zoology and microbiology, do you have anything to get from stem cell, or is your study relates stem cell anywhere in your syllabus? Yes, they do. Because for the botany people, the plant people, you have, I mean, you better know, I don't know that much, but at least I know this thing, that meristems, these meristems have cells which divide many times, and their descendants make up all the new tissues and organ, and that actually is the definition of stem cells. So botany people, you should have an interest in stem cells, of course. What about the zoology people? Yes, of course, because animal and additionally human chordates, they have their own stem cells. Okay, so there is a difference between plant and animal stem cells, but they have something in common as well. What about the division of microbiology? Microbiology is a very concerned subject. You know, right now we all are going through something which is also another microbe, and the major source of contamination in stem cell studies. Now, the purpose of stem cell is you want to transfer stem cells to patient. You want to study how different diseases evolve using stem cells. And during all these things, there is always a chance of getting the cells contaminated. Contaminated by whom? Microbes, bacteria, fungi, yeast, virus, whatever it is. So microbiologists also should know about stem cell, and these microbes actually use stem cells as a very nice thing for the host pathogen interaction. So that is also important for the people of microbiology as well. And another most important thing, that why we are having this online webinar, I mean this thing, because we are all sitting at home because of an issue called COVID-19 pandemic. Does it has something to do with stem cell? It also has. You know, there's mesenchymal stem cells. are very important part have been, and this has been studying, and a lot of clinical trials has already happened till the time this pandemic has started. Why? Because mesenchymal stem cells have the inherent property of becoming, immunomodulatory, and they can restrict the surge of this this interleukin, which is produced when this virus infects. And one most important thing is the entry point of virus into the respiratory system is through this receptor called ACE2, okay, angiotensin converting enzyme 2. So mesenchymal stem cells, interestingly, are devoid of this receptor. So what does it mean? If you put the stem cells inside the body and this specifically differentiate towards the respiratory system, then if the virus itself doesn't found a receptor, how it was going to bind? So stem cells also has implication in this pandemic time also. So what are stem cells? Stem cells are undifferentiated cells that can differentiate into any specialized cells and can divide to produce more of its own kind. So if you think this is the stem cell, they undergo asymmetric division, and then one set can form the differentiated cells and another set forms the stem cell itself. Now this kind of division is a part of adult stem cells. I will come to those things, what are adult stem cells and other. But generally stem cell will follow either this kind of division or this kind of division or together. But as a whole, you can say asymmetric cell division. Stem cells and differentiated cells are generated from a particular stem cell. Now, I should make you understand that what is differentiated and what is undifferentiated. To tell you this in a very lucid manner, I thought of using myself as an example, like see how. So this is me in 2008, I was a postdoc, okay. So at that time, I had only one intention or one thinking in my mind. It is about me only. When I will get a paper, when I will get a job, when every time it was me, thinking about nobody else. So I was a differentiated person. Differentiated means I'm thinking in only one direction, only about a single thing, and that is me only. Then what happened is, when I joined as a scientist, faculty in RGCV, I am this. And this means undifferentiated. Why? Undifferentiated is the stage where now I can't think only of myself. I have to think about, I am part of administration, I am part of different research programs, I am part of a master degree program. I have to think about my PhD students, their future, their careers as well. So I am doing multiple jobs. And that multiple job is undifferentiated. I am not only in one direction. I am working on different direction. So this is the difference between a differentiated cell and an undifferentiated cell. I just represented myself as an example to you. Okay. So what are the properties of stem cells? First is non-committed. They cannot be committed to any particular lineage or lineage. I hope you all know lineage is like the ectoderm, endoderm, and mesoderm. These are the lineages from where different organs of the body generate. So they should be non-committed. On the fun side, I would say just like the political parties, they're non-committed. So if you put some more money, they will become in any other state. Okay, so that kind of non-committed. The second one is self-renewal. That is the most important part is they will undergo numerous cell division while maintaining the undifferentiated. I just now talked about what is undifferentiated state. Third part is the potency, the capacity to differentiate into specialized cell types. Now, this is not same for all the different kinds of stem cells we study. And that is divided into 4 types. First is the totipotent. Totipotent stem cells they can differentiate into all types of specialized cells of the body, plus that is needed during the development. Basically, the connection between the mother and the fetus, the placenta, yolk sac, and umbilical cord. So the zygote, which is formed from a sperm and a ovum, the first one-cell zygote actually is the totipotent in nature because it can give rise to the entire body along with the connection between the mother and the fetus. The second one is the embryonic stem cell, but it cannot form the placenta, the yolk sac, or the umbilical cord. Okay, so it has started when you reach from totipotent to pluripotent. Next comes multipotent. These are the adult stem cells. So where do these stem cells are? Adult stem cells are actually present in your body after your birth. Like your intestine will have a part of intestinal stem cell. your blood inside the bone, there will be hematopoietic stem cells which will generate blood cells. Then you have muscular. Then everywhere you have nervous stem cells, neural stem cells. So there you have this kind of pools of stem cells in each organs of the body, which upon the signal they will differentiate into particular cell types. Like for the blood, from the hematopoietic stem cells, you get red blood cell, you get erythrocyte, lymphocyte, whatever, the platelets, everything. So these are multipotent. They cannot form this hematopoietic stem cell or the intestinal stem cell cannot form back into another type of stem cells. So they are more committed. Okay. Next comes with the unipotent. They can only differentiate into one type. For example, the spermatogonal stem cells can only form sperms. So if you consider this potency of the stem cells, you can think about a plateau which was designed by a scientist, or he called it landscape, Conrad Weddington. So this is the potential or the development potential of the different kind of stem cells. So this is the ball at the top of the plateau. This is totipotent. It comes down to a stage which is pluripotent. So it can go down to this levels, but it cannot go up. So these are pluripotent, and that is the embryonic stem cells. Then comes this kind of red balls. They are the multipotent cells. They cannot go up, but they can go down to this many types. And lastly comes these balls, which are at the edge only, and they can only go down, but they cannot go up. So they are the unipotent stem cells. Okay. So the major types of stem cells are embryonic stem cells, embryonic germ cells, and the adult stem cells. Now today I will be, since the topic of my talk is actually induced pluripotent stem cells, and they replicate, or they are similar to embryonic stem cells. These are pluripotent, can be differentiated to any lineages. Lineages means ectoderm, endoderm, and mesoderm, and hence they are the one who can be used in regenerative biology. Okay. So what are the uses of stem cell research and why it is important for us? Because they can replace diseased or damaged cells. They can differentiate into different lineages, just now I talked about. They allow us to study development and genetics, and they can be used to test different substances, drugs or chemicals, which will be beneficial to the mankind. So these are the purposes which stem cell research serve for. So coming back to embryonic stem cells, the similarity between induced pluripotent stem cells. So it was discovered early in 1981, and you could see this is the one cell stage. This is zona pellucida. It protects kind of the one cell stage, the zygote, which in small term, which is very lucid term, we call zygote. Then it undergoes different cleavages, 2 cell, 4 cell, 8 cell, becomes a compacted morula, and then form blastocyst. So we all, this is a mouse picture, but we all, a human, everybody goes through this kind of divisions when we are inside the mother's womb. I mean the uterine oviducts. So this is a 3.5-day-old mouse embryo, and this particular kind of bulge is called the inner cell mass, whereas the thin bordering line is called the trophectoderm. So this inner cell mass is actually responsible for the generation of embryonic stem cells. And you all remember that this is the area, this is the cell which all of you are made up of. So you are a result of the differentiation of all the embryonic stem cells present in the inner cell mass when you were an embryo. Okay, and the trophectoderm actually gives rise to the connection between you and your mother. Whether it's placenta, whether it's umbilical cord, whether it is yolk sac, everything was formed by this trophectoderm. And I can show you a video where this is a human embryo. This is from a 32 cell stage division, and then when the embryo comes out, and we call it hatching. When the embryo breaks the zona pellucida and it comes out, we call it hatching. And since this is an in vitro, I mean, in the lab it was going on, so of course there is no mother's womb or uterine horn. But generally what happens is, when there is the hatching, the embryo comes and then just gets adhered to the uterine wall of the mother's womb. So let's just watch this video for a few seconds. So this is the division which is going on. This is from morula. Then you get the blastocyst. So this is the inner cell mass, which we are calling. Then so this is the same thing which happens in vivo, I mean inside the body of any mammal or any. So when it comes out, this is the hatching. And in real, this hatched embryo then gets adhered to the uterine wall. Okay. So then you have yes cell, embryonic stem cells. Then why you need iPS cells? Because of ethics concerned with it. Because to get human embryonic stem cell, you have to destroy an embryo. Now, it's upon the wish of the person, whether the person wants to share her embryo or not, which will be obviously very rare. But to avoid this ethical issue, scientists wanted to have something different which can replace embryonic stem cells. Now, in the early years, in 1958, Sir John B. Gordon actually demonstrated that the nuclear transplant of somatic cell, nuclear transplant of somatic cell means you take a somatic cell, I mean your body cell, and you take the nucleus from the ovum, and then you put it inside the somatic cell, and you get an entire organism. So that's the way he produced adult frog, and then there was Dolly the ship, which was also produced like that, and that was called somatic cell nuclear transfer. But it was good for reproductive therapeutic cloning, but it's not as a source of cells that will be transferred from one person, I mean, that will be produced in the laboratory and then get it back into the patient or the person who needs it. So people still started looking for other options for embryonic stem cells. By the time, in 2006, Professor Shin Yamanaka from Japan, he and his lab, actually his team, find out that transcription factors, transcription factors, in short, these are actually proteins, they bind to DNA, okay, and can modulate the transcription of genes. So he found that the transcription factors are actually the factors responsible for getting or reprogramming some cells, okay, and that was the introduction of induced pluripotent stem cell. So you can understand the meaning here. Induce, you're forcing something to become pluripotent, and that's why it's induced pluripotent stem cell, and in short, iPSCs, you can tell. Okay, so transcription factor is a very important thing in generating induced pluripotent stem cells. And these iPS cells are similar to embryonic stem cells. I'm telling similar, I'm not telling equal. Most of you who have maths in your earlier days, you have something that there is a sign called similar and there is a sign called congruent. This two triangles are congruent and two triangles are similar doesn't mean the same thing. So it's not a congruency type, this is similar because characteristically there are difference between iPS cells and the embryonic stem cells because of their origin, from where they're coming. iPS cells come from differentiated cells, whereas embryonic stem cells are coming from originally from the embryo. So there will be, of course, some differences. So this was, I showed you, differentiated, undifferentiated. So this differentiated to undifferentiated induction is actually induced pluripotent stem cells. So why induced pluripotent stem cells? The first thing I told you is to avoid ethics, bioethics. There would be no destruction of human embryo. Next is very important, isogenic cells. That means your own cells will be given back to you. Because if you have heard about, I don't know, there is graft versus host rejection. Like there is always a rejection kind of thing in us. If your parents tell you, do this, do that, you will first inform me. First thing is, I won't do that. Your teacher said, read this, read that. Don't spend your time doing WhatsApp and all this thing. Your first thing is, no, I will do it. So it's a rejection kind of phenomenon is always there with us. So the body also is like that, that if you want to give cells from somebody else, we will reject it. Our immune system is made up like that. We reject like that. Okay. But iPS is what? You will create this pluripotent stem cell from your own body. You differentiate them in the laboratory and you give it back to the same person. So your body will not reject your own cells. So that is isogenic cells. And of course, then another advantage is easier to create because you are not destroying anything involved. It's not too much ethics involved, not too much laborious process involved, but there are hurdles which we will be talking about. So this is the first report of induction in pluripotency. Shin Yamanaka and his postdoc Takahashi, they reported the generation of induced pluripotent stem cells from mouse embryonic and adult fibroblast Fibroblast cells are very, they are the one which is responsible for the secretion of extracellular matrix, the collagen. They mostly lie with the epithelium and the endothelial. They line those parts. And that's why the skin fibroblasts or the dermal fibroblasts, which we call, are the most easiest access or easiest source of generation of iPSCs. So this was reported in 2006 and 2007. And this may be for the first time or something, that the Yamanaka got, along with Sir John B. Gordon, the Nobel in Physiology and Medicine in 2012. So within 6 years, they got the Nobel Prize, which was tremendous. So you can understand the importance, the impact these iPSCs have on the scientific field, and of course the medicine also. So what are iPSCs? These are adult somatic cells, not embryo, okay? Adult somatic cells means adult body cells that reprogram to become pluripotent. Pluripotent means they will generate all the different lineages of the body, ectoderm, endoderm, and mesoderm. How that is done? Exogenous expression. Exogenous means something you are putting from outside of transcription factor. So this is the set which originally Shin Yamanaka introduced. MYC, SOX2, OCT4, and KLF4. Then of course there was some kind of modification when another group used, instead of KLF4, they used Lin28. Lin28 is an RNA binding protein that actually is responsible for the microRNA regulation. And there are some other people who replace c-MYC with I-MYC because c-MYC is actually been oncogenic. So after this expression of exogenous transcription factor, but how do you express them? You can't just put the factor like this DNA, pushing this into the, in the cell. That's not possible. You need a vehicle, okay? And the vehicle is the viral vectors, mostly. So retrovirus, lentivirus, episome, sendai. So see, microbiologists, you have a big role to play over here, because retrovirus as such or lentiviral as such, they are not actually good for you, us, nobody. But the virus has been modulated or manipulated in a manner so that when they are put into the system, this mammalian cells, they are no more having viral gene expression. Rather, they will express the gene like this genes, OCT4, KLF4, SOX2, into the system with the minimal viral proteins required for its structural formation, that being retained. So those things have been taken care. Episome is just like a plasmid. Plasmid, it's a DNA which is there in bacterial cells, and episome is also an entity of DNA which can live on its own, and it will express protein. And Sendai virus are the most new one and has been found to be the maximally effective and very good also in a case that it doesn't enter. Sendai is also an RNA virus. It doesn't enter the nucleus. What it will be doing is into the cytosol. So within 10 passages of cell culture, this virus will be washed out from the iPSCs being produced. So it is much more ethically correct to use Sendai virus for the transfection, for the generation of induced pluripotent stem cells, if its purpose is to give it back to human being. Okay. And sometimes small molecules like some inhibitors, activators kind of things are added to manipulate or induce the formation of iPSCs. So the steps involved in practically, how do you do the experiment? First is transfection. Transfection, I don't know whether you all are acquainted with this term. Transfection is giving the DNA, supplying the DNA into a mammalian cell with the help of any vehicle. Here we are talking of vector as viral vectors at the vehicle, but in other things, lipid mediator, other things are also there. So transfection of these cells, and the cells may be fibroblast, adult fibroblast. The vehicle being used is retrovirus or lentivirus or episome, which I talked about. So after 3 to 4 weeks, you will see the small transfected cells become morphologically and biochemically similar to pluripotent stem cells. So how do they look like? This is the way they look like. So see this, these are the surrounding cells, which are the fibroblasts. At day 7, this cell starts coming closer to each other. By day 10, they become more compact. By day 12, they're highly compacted, and that is the formation of iPSCs or induced pluripotent stem cell. At that point, what you do, you take out those things manually with a tip, and then you break them into single cell. Those who are doing cell culture, they will know the trypsin is an enzyme which will disintegrate this colony into single cell, and this then single cell will be actually responsible for the formation of one big colonies, and they in the simultaneous passages would look like this. So nanog is again a transcription factor which is associated with pluripotency, and they express. So you know that yes, you have produced an iPSC which is expressing pluripotent genes as well. So this, in short, is the application of iPSC. I will just tell one by one. So suppose this is the patient for whom we have to generate some cells, I mean some stem cells, which we have to give it back to that person. So we will take either keratinocytes, keratinocyte is again from the skin, then fibroblast, or even peripheral blood mononuclear cells. They are very good source of generation of iPSCs. So you get these cells here, then convert them into pluripotent one, I mean iPSCs, with the expression of the transcription factors and with the vectors like Sendai virus, simple plasmids, or mRNA, and you get iPSCs. Now you can do two types of things. You can directly differentiate them, forming all the three lineages. Beta cells is endoderm, cardiomyocytes is mesoderm, motor neurons ectoderm. Or what you can do is you can do genome editing, because this person was already suffering. Okay, suppose Alzheimer's disease, they have problem in the DNA itself. So you need to correct that genome, and there are a lot of tools now available for correcting the genome like CRISPR, TALEN, zinc finger. The gene correction is done, and you have a genome edited induced pluripotent stem cell. That means a correct one, and you can differentiate them, and then you can put it back to the patient. Or with the uncorrected one, you can just simply do drug screening or disease modeling. Disease modeling is when you will develop a disease in the lab, in the tissue culture lab. This is disease modeling, because you want to study what are the factors which is helping this disease, what are factors which can inhibit the disease, what are the other things which I can add or subtract and can help in getting rid of the disease. And the corrected one can be used for stem cell therapy and give it back to the patient. So these are the way this iPSCs has its application, and it's tremendous application. So how was it proven? So this is the concept that bed is the patient. You take the cells out in the bench work, lab work, and then again you give it back to the patient, and that is why it's called bed to bench to bed again. So this was a proof of concept which was done as early as 2007. Okay. So what was the proof of concept? Proof of concept is, suppose you hypothesize something and you think that this will work, and that will work for a higher, higher system. Higher system is, here I am talking about human. No, you cannot do all the experiment in a human. So that's why you need the lower animals, the mouse, the rat, then the rabbit. That's why. So the proof of concept is generally done in the smaller animals to go to the level of human. So what they did is they prepared a humanized sickle cell anemia. I think all of you heard about this sickle cell anemia mouse model. So we have mouse model of different kinds of disease available, and they are in huge stocks in different kinds of repository like Jackson, like Charles River. They're all in US. I wish these things are also available in India, but there is a trial in ISER Pune, I think, that's been going on. Okay. So this humanized sickle cell anemia model was created, and they harvested the tip fibroblast, and the fibroblast was then transfected with this OCT4 SOX2 KLF4, and iPSCs were generated. Then it was corrected by gene targeting, the beta sickle mutation, you all know, then differentiated and put back into the mouse, and the transplant corrected the hematopoietic progenitors back into the irradiated mice. So this was the proof of concept that yeah, it will work. Then people started immediately doing disease modeling of Alzheimer, Parkinson, Huntington, because these have no medicine to care about. But what was the success? It took 8 years to get the first success of using iPSCs for a patient to finally get rid of a disease. And that was in 2014, when a Japanese woman received her own retinal sheath tissues being prepared from her own stem cells, skin cells prepared, and she was suffering from acute macular degeneration, and it was implanted into her eye, and till now, till date, she is doing fine. So the iPSCs were generated by, again, Shin Yamanaka in his university, Kyoto University, whereas the surgery was done by Masayo Takahashi at the Riken Center for Developmental Biology. This was the first breakthrough. But you know what, this patient versus one patient generation of eye cells is humongously costly. It's impossible for we Indian to do this one because it's autologous, okay, and it takes really, really big time. So what scientists started thinking is better to be allogenic. Allogenic is like HLA matched those patients or those human beings which have a similar kind of HLA, they can receive cells from one another. So Yamanaka right now is trying to get that bank done, and since the population of Japan is not that much, only 75 iPS cell lines will actually cover 80% of the Japanese population, which is impossible in our country because we have a huge number. So what does it mean? Suppose X person and Y person have similar HLA, okay? So the X person can donate the stem cells being generated from his or her somatic cells and give it to the Y patient. So there is no need of getting cells from Y to give it back to Y. So it saves time, it saves huge amount of money. So that is what is allogenic iPS formation. So till now, this second generation of reprogrammed cells are being done and being put into different from patient X to patient Y, and it is getting successful day by day. But why it took so long? 2006 to 2014, then 17, and all these things. So that means there is some issue with the generation of iPSCs. Actually, yeah, there are a lot of hurdles. From somatic cells to induced pluripotent stem cells, it takes so many hurdles to come. The first problem is uncontrolled growth of the cells, remaining cells. Now, if some cells fail to reprogram into iPSCs and differentiate, then they will become uncontrollable. They will actually form tumor when you put them inside the body, and this is completely impossible. You can't. You just want to cure a person, and you cannot put a cell which the person will then generate a tumor. So people have to take great care of before applying these cells into a human being. Then genomic instability. Now this is a big issue also because generation iPS takes 25, 30 or sometimes 35 days at a time, one time generation, and then passaging, then checking it's good or bad, it's all the characters. So it's a n number of days of continuous culture in the laboratory, and those sometimes lead to genomic instability, and that is again dangerous if you put it back to a patient. And the next, the most important, I would say, is the low efficiency. The efficiency sometimes is 0.0001%, 0.1% is, or 0.2% is the highest one. So with this kind of efficiency, you need certain amount of cells to give it back to the patients. No? So you have to work on these aspects. And all these things, from somatic cells to induced pluripotent stem cells, the basic need is epigenetic remodeling. Okay, this epigenetic remodeling should be done. The cell should be completely reprogrammed, remodeled in the terms of epigenetics. Because when you transform a somatic cell to an induced pluripotent stem cell, there are steps like X chromosome reactivation, DNA demethylation, all this developmental gene expression and downregulation is a matter of fact because of epigenetics. So epigenetics has to be completely done. So one cell will be completely devoid of one character which it was having earlier, and it will gain a new set of character. And this should be done 100% correctly. Then only the reprogram or induced pluripotent stem cell formation would be the perfect one. Epigenetics becomes so important in 2010. The Times magazine actually highlighted epigenetics. And this involves DNA methylation at the cytosine, histone modification, histone modifying enzyme, histone variants, small and long non-coding RNAs. So in short, what is epigenetics? Is the term again was coined by Waddington, and it means epi means beyond or above genetics, which means there is no change in the DNA sequence, but yeah, the expression of gene will be affected, and that's why the character will be affected. That is epigenetics. The two most common ones, the DNA methylation and the histone modification, I will be going straight to the histone part because that will bring into the little bit of work which I am doing in my lab here will introduce you to. Okay. So for that, first you should be knowing what is a chromatin. You all have heard about chromosome. So the next, chromosome is the most compacted part. Next you come down is the chromatin. What it is? It is actually when the DNA is wound around proteins or the basic proteins, histones, thereby forming a complex known as the chromatin. Why you need chromatin in a cell? You know, the DNA inside the nucleus is actually 2 meters long. Neither me or I think most of you students, none of you, your height is 2 meters, I think. So how will a DNA be there in my, it's not even my length. So how to do that? So nature, evolution, whatever you call, it is compacted into the nucleus by the formation of this kind of chromatin structure. So the condensing of the DNA within the nucleus is the purpose for getting to chromatin. Then the control the DNA. So if something is too much exposed, so the reaction will be much more. In chemistry also you learn, no? The more you increase the surface, the rate of reaction and as well as the reaction increases. So if you leave the DNA exposed, it will be degraded pretty faster, no? So the way to control is also to let it form the chromatin. To strengthen the DNA to allow mitosis and of course to avoid DNA damage. Okay. The chromatin structure is the important one when we study epigenetics, and that is the first level structure, the nucleosome. Nucleosome is just that unit where the DNA is wound around that octamer, histone octamer. And it's a very dynamic structure. You know, it opens, closes. And when it is in a closed conformation, we call it heterochromatin. When it is in an open conformation, we call it euchromatin. In our cell, in the nucleus, it is mostly heterochromatin, closed conformation, and very rarely it is euchromatin. So whenever there is need of expression of the gene, it becomes euchromatin. Whenever it is not required, it is heterochromatin or closed or compacted. So this is the way. So if suppose during the pluripotency, you need the genes like nano, OCT4, SOX2. So those promoters, you can say, in the fibroblast, they are in the compacted state. That means they are not getting expressed. So during reprogramming, what happens is the chromatin becomes closed, or it becomes like euchromatin, it becomes exposed so that the transcription factors or whatever these modifications can happen in this stage and the genes get expressed. So this is the octamer histone, which is H2A forms of histone. Histones are basic proteins which is formed from H2A, H2B, H3 and H4. And see this white part, this is the DNA binding around, and H1 is the linker. So these are basically canonical histones. I mean, me, you, everybody is the same. There's no difference. So they don't play any non-regulatory role. They don't play any regulatory roles. They are non-regulatory in nature. But there is something which actually controls or regulates the chromatin, and that is the tail of this histone. See these tails. And these tails mostly have K. K is the lysine amino acid. So this amino acid as well as arginine, phenylalanine, serine, they have a tendency of getting modified. Modified means enzymatically modified. So AC means acetyl group gets added. Me means methyl groups added, then PO4 phosphate groups, protonylation, ubiquitinylation. So this kind of groups being added by different enzyme makes the tail a little bit, bulky, and that makes the chromatin open. So depending upon the marks, the modifications, whether it's acetyl or methyl or phosphorylation or protonylation, it depends whether the chromatin will open up or it will close. So this is for the part of non-regulatory histones. But there are histones also who vary a little bit with their own ancestral protein, like histone H3 and H3.3. So H3.3 is called a histone variant of histone H3 because it has almost everything similar, but there are something different. And this difference, which are called non-canonical histones, make them regulatory. See, they can take part in transcriptional activation, repair, recombination, everything. So they have a regulatory role, histone variants. And these histone variants are incorporated or put up into the chromatin by histone chaperones. Histone variants are very important in pluripotency also, and these are being recruited to the chromatin by histone chaperones. And here comes a little bit of work which I'm going to share in this coming 5 minutes. That what me and my students are working on role of histone chaperones in inducing pluripotency. So histone chaperones, I think you will be all knowing, or few might know that heat shock protein are chaperones. So chaperone in literature says means something which is helping others, okay, as a helping protein. So histone chaperones are broadly defined as protein, of course, and they are responsible for chromatin assembly, disassembly. So we just now talked about chromatin. So this nucleosome being there and or it being evicted or being incorporated, so they will lead to assembly and disassembly of the chromatin. Histone transfer means histone is being synthesized in the cytoplasm and then brought into the nucleus. So histone chaperone do this. And histone exchange, just now I talked about histone variant. So this exchange, incorporation, eviction is done by histone chaperone, which will facilitate DNA replication, repair, and transcription. These chaperones also modulate directly or indirectly histone post-translational modification, and these are the modifications I just now talked about: acetylation, methylation, phosphorylation. All this group when being added into the tails of the canonical histones, they are functionally crucial for the processes like replication, repair, and transcription. So this is the way they function. So what does it mean? It means these are very basic molecule of life. So they will be needed. So why I want to study? They will be needed anyway. But my concern was the efficiency, the time, why this IPS takes so long, so much time, and so many days, or so many kind of barriers are there for requirement. But is any histone chaperone also is there who is acting as a barrier of reprogramming? So we started with the question and saw the expression of mRNA expression. You know, the DNA transcription to mRNA and mRNA again translated to protein. So mRNA expression actually is the gene expression we talk about, and mRNA are converted into cDNA, okay, complementary DNA by reverse transcriptase enzyme, and those we take as the expression of gene at the RNA level. So when we looked into the differentiated cell fibroblast versus the embryonic stem cells, why? Because they are similar to iPSCs. So as a starting material, we wanted to see the expression of these histone chaperones in the fibroblast versus differentiated versus the undifferentiated cells. We found out, look at this dotted one, a histone chaperone called aprotixin PNK-like factor was very less significantly, which in scientific terms we call significantly less in embryonic stem cells. That means the pluripotent cells, whereas it was huge in the differentiated fibroblast. What does it mean? So we just looked into the different stages or days of reprogramming. Now see, this is days of reprogramming: 0 day, 3 day, 5 day, and finally formation of iPSCs. We found at the protein level, this is Western blot. So these are the protein, APLF protein. We found that APLF was very low, and then finally in iPS present in the differentiated, you see almost no expression. So that means it is present differentiated in a very nicely manner, and it goes down when we thought and we hypothesized that if you reduce the level of this APLF in the differentiated cell, then will it be really easy to get the iPSCs? And then we did knockdown of APLF in the fibroblast, and we found, yes, the efficiency, that means the formation of iPSC colonies, increased from 0.01 in the control cells to the knockdown cells 0.25%. So it is around 25 times increase. And the time was reduced to half. So half the day, 10 days were required for the formation of iPSC when you lower the expression of APLF in the cells than when the APLF expression is not lowered. So this is the significant result we find out, okay? And when you get this iPSCs, you have to tell that, yes, you have got it. How do? You have to prove that these cells have the pluripotent character. So there are n number of in vitro, laboratory related experiment. Then you have in vivo in the mouse related experiment, which is required to show the full potential of the cells which you have generated. But I would just like to show you one which seems very interesting to me. We differentiated these iPS cells to beating foci. I mean, it almost seems like heart is beating. So this is done. Just look at this area which I'm rounding with the mouse. So it's beating. So this cells in the lab itself, in the tissue culture dish, generated this beating foci. So it showed that iPS cells have the quality or the potential to differentiate. Now, you have to finally say that how it worked as to barrier to reprogramming. So that is called mechanism, how it is working. When I showed you that from a differentiated cell to iPSC cells, there are different stages like X chromosome inactivation, development gene expression. There is a first step I showed is MET, which is called mesenchymal to epithelial transition. So this is a change in the phenotype of cell from mesenchyme type to epithelial type, and it's a very common phenomena, both this and the opposite of this, EMT, in all the different diseases, development, everywhere it is. And the epithelial gene which is responsible this is the E-cadherin CDH1. So we found that when APLF, the blue one is APLF is present, it interacts with its histone variant macro H2A.1, which is a repressive histone variant, and gets itself recruited at the chromatin of or the promoter of CDH1, and that leads to the compaction of the CDH1 promoter and leads to the reduced expression of CDH1. Another thing we found as at the pluripotent genes, NANOG and KLF4 promoter, when the APLF interacts with the macro H2A1, it inhibits the incorporation of H3K4 dimethylation. Now, this is a histone modification and an active one. Means when it is present, the chromatin will be or the promoter will be in relaxed or in a open condition and the gene will be expressed. But because of presence of APLF, it is very, very less, and that leads to the compaction of the chromatin or the promoter and less expression of NANOG and KLF4. So when you knock down or when you reduce the level of APLF, what happens? There is considerably less APLF and macro H2A1 here, and that's why the chromatin or the promoter remains relaxed and CDH1 expression is induced. And here also, when there is APLF less expression or reduction in expression, there is more incorporation of H3K4Me2, and that leads to the open chromatin and the induced expression of NANOG and KLF4. So mechanistically, this is the way APLF worked, and it acts as a barrier to reprogramming. When you lower the expression significantly, you downregulate the expression of APLF, the induction in pluripotency is enhanced, both in efficiency and in time reduced also. So this work we published in Journal of Cell Science, and this is the basic go to home message is like iPS is nothing, it's a biological clock is being turned opposite. So we started from here, our life started from here, early embryo, then that stem cells, embryonic stem cells from here giving rise to your all lineage committed cells, then all the adult stems in the different parts of or different organs of your body. This then leads to the formation of the mature body cells, and when you pick up this mature body cells and reprogram them with the transcription factors, you get induced pluripotent stem cell, and ideally these induced pluripotent cells can be driven back to the embryo. This is unethical, but we have restricted ourselves in generating the differentiated cells from the iPSCs and put it back to those humans who need them and for their survival. So I will end here, and I would like to thank, of course, Dr. Tamal for inviting me to present this webinar. And next is our institute where I'm doing the work. This is our director, Professor M. Radhakrishna Pillai, who just retired, but was an immense support to me and my lab. My students, these are only 4 here because we are now during the pandemic, we are doing staggered lab setting. So 4 comes in the morning, 3 in the afternoon, kind of this. Only 4 were available at that time. But the work was actually done by Khaja Mohidin's side. So he is right now in Raiken. Sunu is in CDFD, she is SRF now, and Aditi is the one who joined the University of Wisconsin as the postdoctoral fellow. So thank you very much for listening to the talk, and before I wind up, I would just take 1 minute time to tell you that since all of you are doing bachelor's degree here, and I think most of you will be also interested in doing the master degree, RGCB has started this unique MSc program in biotechnology. It's mostly based on practical oriented teaching. So we are keeping students mostly in the lab and learning from there rather than theoretical sitting in the class and studying. And we are actually offering this disease biology a specialization course, which is nowhere being offered in India at all. Other specialization being genetic engineering and molecular diagnostic and DNA profiling, because molecular diagnostic is a very big part of RGCB, because I don't know whether you know or not, but ICMR, I mean, according to ICMR, RGCB is one of the COVID testing center, and we have developed our own kits also. So we are good in molecular diagnostic and this thing. And anybody from any field, I mean, we have kept it open for pure science, for biological science, for pharmacy, physics, chemistry, any stream are open to join this MSc Biotechnology program, because we believe in interdisciplinary science and interdisciplinary biology. So thank you, and I would be happy to have your questions. You can ask the question any language you feel comfortable with me, Hindi, English, Bengali, anything. So over to you, Tamal.
Thank you, Dr. Datta. That was magnificent. It was superb of a presentation. We really got amazed to the kind of work you are doing, the kind of thought that modern scientists in India have brought in. That is really it's something to look up to. We are really amazed with that. Certainly, my students, our students rather, they can have a few queries. So, students or any participant, if you have any query, please don't hesitate to ask. You can unmute yourself, or you can type it in the chat box so that ma'am or us can read the questions and address your problems. So the discussion is open for the talk is open for discussion, you can say.
I'm sorry, I might be too fast a bit, but there is time limit 3 to 4, so I was seeing the watch like it's going to be 4, so I just made it a bit faster.
No, that is fine. That is excellent. It could have been much better if you had focused on your own research a little bit more. I hope that will elaborate with time. Certainly, with time that will elaborate. So next time you will be here, so we will listen more on your research. Yeah.
I actually didn't do it because I thought students will be more there and to go into
exactly. Yeah. For undergraduate platform, yeah, that is good one.
I avoided that part very intentionally actually.
Okay, and exactly, on the way you integrated all the students from botany and microbiology as well, along with the zoology students, that was really fantastic. So students, please put forward your queries to ma'am.
Or if anybody is feeling shy or something like that, you can just send me an email also, no problem.
You can also ask the questions on the chat box. You can type your questions on chat box. Yeah. By the time they break up their silence, I ask a few questions. Would you mind?
No, not at all. Yeah, please.
Okay. Yeah. That experiment by Professor Yamanaka and his team. From that experiment, we came to know about the different transcription factors that help in the process of de-differentiation or reprogramming, you can say, of the somatic cells. So sets of transcription factors were used like CMYC, SOX2, and KLF, all these things. What was the strategy exactly for choosing such transcription factors? Because we know that there are plenty of transcription factors in the whole development process.
Yeah, absolutely. So what they did is see that embryonic stem cell was discovered quite early, 1981. Then 1990 it was human embryonic stem cell. So that time they people knew that what are the genes which are responsible for pluripotency. They know, but that is also a big network of lot of genes being responsible for pluripotency. So what they started actually with 100 transcription factors. The start was from 100. Then they checked each one of them. Then they came down to 20. With the 20, they did combination. I mean, 1 + 1, 1 + 3, 1 + 4, or enormous number of combinations. And this combination.
That was the whole process of standardization.
Exactly.
That was outcome of the whole process of standardization. It was not randomly chosen, or it is not strategically chosen. Okay. So the best results were for those. Yeah. for those transcription factors. And one more question from your research, that you are more focusing on the transcription of the Histone chaperones, like APLF, as you told. You could be able to downregulate the expression of the APLF, and in a much faster way, in a temporal fashion. I want to know, maybe I missed out, what was the factor, means what was the thing that you had induced the cells with, so that this APLF downregulation could be so faster?
No, APLF downregulation was not faster. APLF downregulation led to the faster induction in pluripotency.
Okay, means then what was the agent actually that brought down this downregulation of the APLF?
Oh, shRNA, lentiviral mediated shRNA.
Okay. Any other queries?
Dr. Tamal. Dr. Tamal, myself, Dr. Shaurish.
Yeah, Shaurish sir. Yeah. Shaurish sir from the Department of Botany. He is there to ask some queries, I guess. Yeah. Shaurish sir, please.
Ma'am, I have a basic question. First of all, thank you for your beautiful presentation. I have a very basic question to you. So can you use, you talked about the retroviral transfer or viral particle as vectors of transferring the stem cells, embryonic stem cells. So can we use the bacterial plasmids or yeast plasmids for that? Or is there any way?
Not bacterial
just to transfer.
No, because you need all the mammalian related things, no? These are all mammalian cells.
Okay,
and the efficiency, why this viral, people have moved to viral and all these things, is they are getting integrated into the genome, is a very positive, as well as the disadvantages you can also tell, because if there is some leaky in integration, the mammalian genome will be completely disrupted. But the efficiency of getting into this one, I mean, even if you're using these viral vectors, the efficiency is 0.1%. You can think about other, so it will be disastrous. So that's why.
Okay, so expression is better. That is why we are using.
Yeah, expression is better, and of course, mammalian. The system is mammalian.
Oh, thank you, ma'am.
Thank you, sir.
Thank you, Shaurish sir. Any other queries? If not, then I shall like to ask Priyanka ma'am to do the needful, to thank the speaker from the departmental point of view and the institutional perspective as well. Some queries are there? Okay.
Provides a hint for the success of stem cell therapies. My question is, ma'am, can you elaborate on that? Provides a hint for the success of stem cell.
That stem cell based therapy is of COVID-19, as you told, that mesenchyme lag,
mesenchymal stem cell. Mesenchymal stem cells are being used because of their character, their nature, being immunomodulatory, and they inhibit the surge of release of this, basically TNF alpha. It is being hugely released when this virus infects. And the additional advantage is its absence of the receptor which the virus spike protein actually binds to when it enters first into the respiratory tract. ACE2, it lacks, the mesenchymal stem cells lack. Okay, Dr. Tamal?
Yeah, I don't find any more queries. Okay. Just
Thank
hello?
you, Shan. Yeah.
Hello, Ananna ma'am. Hello. Hello, Ananya ma'am.
Hello.
Yeah, it's over to you now. The presentation is over, and the queries and clearing is also done to some extent. Most of the students they just felt shy. I think I can understand. And anyway, that was a fantastic presentation from ma'am, and a big thank from the Department of Zoology. So it's over to you, Ananya ma'am, now.
Okay. Thank you, ma'am. Thank you, ma'am, for your presentation.
Thank you.
I would like to ask Priyanka, ma'am, to please give the vote of thanks.
Thank you for giving me the opportunity to express my gratitude today. I would first like to acknowledge the support provided by Father Principal Dr. Lalit Tirke, who always encourage us to conduct such programs. Thank you, Father. I would also like to thank our speaker of the day, Dr. Debashri Datta, for such an captivating speech. I'm sure your speech has enlightened our student immensely. Thank you so much, ma'am.
Thank you.
I take immense pleasure to thank my departmental colleagues. It would not run so smoothly without the support of you all within this short period of time. Thank you all. I extend my thanks to my colleagues from other disciplines like Department of Microbiology, Department of Botany for your gracious presence and your support. Finally, the most important corner of the institute, my beloved students, I would like to thank you all for being a patient learner and for your active participation. I further extend my thank if I missed anyone in my list. Thank you all.